Hello guys, welcome to this YouTube channel. In this video, I am going to discuss the answer key to the Biochemistry section of GATE XL 2024 paper. Let us get started. So the first question is, which of the following pairs of amino acids is not incorporated in a polypeptide chain? And the answer is C, which is ornithine and citrulline. This is because, first of all, there are no codons for these two amino acids, and they are mainly intermediates in biosynthetic pathways such as urea cycle, and hence they are not incorporated in a polypeptide chain. However, if you look at the other options like hydroxyproline or gamma carboxyglutamate or even hydroxylysine or desmosine, these are mostly post-translational modifications that are present in a polypeptide chain, and hence ornithine and citrulline as such are not incorporated in a polypeptide chain. Moving on to the next question. Mammalian cells cultured at low temperature, that is 25 to 30 degrees Celsius, leads to an increased sterol content in the membrane. Elevated sterols in the membrane results in, the option is, an enhancement in the membrane fluidity. Why? Usually, if the sterol content is high at high temperatures, if the sterol content is high, like cholesterol content is high, then it results in increased rigidity and less fluidity. But here it clearly states that there is low temperature, which means there is an opposite effect, which means there is a decrease in rigidity at low temperature and hence an increase in fluidity. So at lower temperature, if you culture mammalian cells, the elevated sterols will result in an enhancement in the membrane fluidity. Moving on to the next question. Which one of the following metabolic intermediates is common to glycolysis, nucleotide synthesis, and glycogen synthesis? The option is C. This is a pretty easy question because we all know the basic biochemistry pathways that we study, that we have in our curriculum. And if you look at glycolysis, the first step for glycolysis, the first step is conversion of glucose to glucose 6-phosphate. So glucose 6-phosphate is involved in glycolysis. If you look at nucleotide synthesis, especially the HMP shunt or the pentose phosphate pathway, here also glucose 6-phosphate gets converted to 6-phosphogluconolactone. And even in glycogen synthesis, glucose 6-phosphate gets converted to glucose 1-phosphate. And hence this glucose 6-phosphate is common to all three, that is glycolysis, nucleotide synthesis, and even glycogen synthesis. And hence the answer is option C. Moving on to the next question. In mammals, hematopoietic stem cells, that's basically HSCs, that give rise to different types of blood cells are known as, and the option is answer B, which is pluripotent stem cells. Why? Because hematopoietic stem cells, they give rise to both myeloid and lymphoid progenitor cells, and totipotent stem cells also give rise to extra embryonic structures, which is not the case for hematopoietic stem cells. They are pluripotent stem cells because they give rise to cells of different lineages and they constantly keep building the blood system. So that's a pretty easy question to score. Moving on to the next question. Which one of the following statements correctly describes the addition of N nucleotides during the rearrangement of the immunoglobulin heavy chain encoding gene? So first of all, N nucleotides are involved, which means the enzyme that catalyzes this addition of N nucleotides is TDT, that is terminal deoxynucleotidyl transferase. So hence option B is correct, and it's a lymphoid specific enzyme. And if you read about the VDJ recombination theory, the variable region mainly involves this VDJ recombination, and hence this addition of N nucleotides is very common in the VD and the DJ junction, and hence option C is also correct. So if you're quite thorough with immunology concepts, these are pretty easy to score. So the answer is option B and C. So this is a multi-select question. So we have to be really careful about choosing the right answer in order to get the full score. Moving on to the next question. A newly identified viral protein contains one long alpha helix spanning 60 amino acids residues, and so the number of main chain hydrogen bonds formed in this helix is what? So this is an integer type question. So you have 60 amino acids. I'm writing AAs for amino acid. So for an alpha helix, which is basically a secondary structure in a protein, so in an alpha helix, the CO group of one amino acid forms a hydrogen bond with the NH group of the amino acid that is 4 residues away. This fact has to be kept in mind when you solve questions related to alpha helix. So in that case, you will have the first amino acid, the CO group of the first amino acid, forming hydrogen bond with the fifth amino acid, the NH group of the fifth amino acid. Similarly, the 2 forms with 6, 3 forms with 7, and it goes on until 56 forms a hydrogen bond with 60. The last 4 basically cannot form hydrogen bonds, they are left out, and hence the total number of main chain hydrogen bonds formed in this helix will be 56. So if you know this particular fact, then you are sure short of getting the full score for this NAT, that is numerical answer type question. Moving on to the next question. In a lactic acid solution, the pH is 4.8. The concentrations of lactic acid and lactate are 0.01 molar and 0.087 molar respectively. So the calculated pKa of lactic acid is what? So this is a basic chemistry question wherein you use this Henderson-Hasselbalch equation, which is pH equal to pKa plus log salt by acid. So which is lactate, concentration of lactate divided by concentration of lactic acid. So that is going to be the pH we know from the question, which is 4.8. 4.8 equal to pKa, which we are supposed to find, plus log of the concentration of lactate is 0.087 divided by 0.01. So that gives you 4.8 equal to pKa plus log 8.7. So if you look at log 8.7, so 4.8 equal to, if you take the logarithm of 8.7, you get the answer to be 0.94. And hence, if you rearrange pKa equal to 4.8 minus 0.94, you get the answer to be 3.86. So the accepted range in the answer key is 3.8 to 3.9, and they have asked to round it off to 1 decimal place, so I would round it off to 3.9. So you get a good score here. If you know this particular equation, which is pH is equal to pKa plus log of salt by acid, then it's an easy question. Moving on to the next question. If a 10 millimolar solution of a biomolecule in a cuvette of path length 10 millimeter absorbs 90% of this incident light at 280 nanometer, the molar extinction coefficient of the biomolecule at this wavelength is what? Molar inverse, centimeter inverse. So here you should know the Beer-Lambert's law, that is A is equal to epsilon Cl. Okay, A equal to epsilon Cl. And A is absorbance. Epsilon is the molar extinction coefficient. C is the concentration and L is the path length of the cuvette. Okay, so now how do you calculate absorbance? Absorbance is given by the formula log of I naught by I, where I naught is equal to the intensity of the incident light. Incident light intensity, I equal to intensity of the transmitted light. Okay, so now we know that 90% of the incident light is absorbed, which means I equal to I naught divided by 10, right? Because if 90% is involved, 10% is transmitted. So now, feeding all these values in the equation, you will get log of I naught divided by I naught by 10, which is equal to epsilon, which we are supposed to find, into the concentration. What is the concentration? Which is 10 millimolar. Since they have asked the epsilon value in molar inverse, centimeter inverse, it's important to feed all these values in those units. 10 millimolar is 10 into 10 power minus 3 molar into the path length is 10 millimeter, which is 10 into 10 power minus 1, right? So now if you take the logarithm, which is log 10, which is 1, is equal, I naught by 10 is 10, so log 10 equal to 1 equal to epsilon into 10 power minus 2. And hence epsilon equal to 1 divided by 10 power minus 2, which is 100 molar inverse centimeter inverse. And the accepted range is between, I think, 95 to 102, and hence the answer is perfectly fine. Okay, so this is a simple Beer-Lambert's equation that you need to apply for getting this question correct. Okay, moving on to the next question. Yes, the metabolic intermediates provide the backbone for the synthesis of amino acids. Match the metabolic intermediates listed in column 1 with their corresponding amino acids given in column 2. So for this question, I will prefer you to know which amino acids are being synthesized from these metabolic intermediates by looking at the different cycles. So here the correct answer is option A, which means alpha ketoglutarate is involved in the synthesis of glutamate. Of course, it's pretty common, it's pretty obvious from the name itself. Ribose 5-phosphate is involved in the synthesis of histidine, and 3-phosphoglycerate is involved in the synthesis of serine, and phosphoenol pyruvate is involved in the synthesis of phenylalanine. So if you can remember these as some facts in your brain, then it's going to be a short question, and I believe this question will have a greater weightage because there's so many facts that you need to remember here. Okay, moving on to the next question. This is a pretty easy question to solve. So which one of the following is the correct match between the molecular properties listed in column 1 and the corresponding biochemical separation methods in column 2? So if you look at solubility, the first thing that comes to your brain is salting out. Okay, and if you look at ionic charge, the first thing amongst all these, the separation based on ionic charge is exploited in the technique of isoelectric focusing. And if you look at polarity, of course, it is used in reverse phase chromatography, because chromatography depends on the relative polarity between the stationary phase and the mobile phase. And of course, the molecular size, it depends on ultracentrifugation. Okay, so the correct option now is C. Okay, so this is a pretty easy question because we would have done a lot of practicals based on this and would have also taken up courses based on various biochemistry techniques. So this should be an easy question to solve. Okay, the next question. Which one or more of the following statements, it's an MSQ, are all correct regarding the electromotive force generated by electron transfer chain? So first of all, it is used for the synthesis of ATP. This is like the first thing that we know. And it's an active transport process, and hence option B is wrong because it's pumping against the gradient, and it includes a pH gradient component. That is perfectly correct because there is H plus transport against the concentration gradient. and it does not include an electrical potential gradient component. That is wrong. So the correct answer is option A and C here. Okay, moving on to the next question. Which one or more of the following statements is or are correct regarding the transport and retention of proteins in different cell organelles? So here it's important to again remember certain facts. So first of all, nano 6-phosphate residues are involved in targeting proteins to lysosomes for degradation. So this option is correct. Transport of proteins into the mitochondrial compartment is aided by, yes, positively charged amino acid residues at the N-terminus and of course internal hydrophobic membranes. So again, this is the correct option. And the retention of protein in the ER lumen requires the K-del sequence motif at the C-terminus. This particular K-del sequence motif, I have seen this in many competitive exams which is being tested, and hence I would request all the viewers to have this particular fact in their mind. And nuclear proteins are transported in an unfolded conformation, and the nuclear localization signal sequence is subsequently cleaved by peptidases in the nucleoplasm. This is a wrong statement. So if you properly read about how the proteins are packaged and transported to different cell organelles, then the correct options are A, B, and C. All right. Moving to the next question. Which one of the following statements correctly describe fluorescence spectroscopy? So when you read about fluorescence spectroscopy, it is not like the normal spectroscopy, which depends on the excitation wavelength. Instead, the correct options are the emission maxima is independent of the excitation wavelength. So this is something that most people make a lot of mistake with. So this is a correct statement. So despite the excitation wavelength, irrespective of the excitation wavelength, the emission maxima is reached. And the emission maxima depends on the concentration of the quencher. No, definitely not. The quencher only decreases the intensity. And the emission maxima varies with solvent polarity. Yes, that's mainly because of the interactions. And the emission maxima varies with temperature. Yes, because mainly fluorescence spectroscopy works with, the electronic and vibration states, and hence this is dependent on the solvent polarity and temperature, but not on the excitation wavelength. So that is the reason why the options A, C, and D are correct. Okay. Moving on to the next question. Which one or more of the following statements is or are correct in the processing of pre-mRNA? Carefully note this, pre-mRNA in eukaryotes. So here the correct options are C and D. I know it's very tempting to tick B because this is 5' capping and 5' capping and 3' poly A tail adding. But this is after the after the mRNA is formed. So splicing happens before that. So splicing, if you look at the basic reaction mechanism, it involves trans-esterification reaction, and of course you have different different patterns of splicing, which is called alternative splicing, and this can of course yield different mRNA products. And of course that's the reason why you have isoforms for a given protein, different isoforms for the same protein sequence, for the same mRNA sequence, forming different types of proteins. So this, of course, takes place during the processing of pre-mRNA, and hence the correct options are C and D. And of course, there's no 3' to 5' exonuclease activity. Yes. Moving on to the next question. Which one or more of the following statements correctly describe the changes upon the addition of puromycin during eukaryotic translation? So first of all, it's very important that you go through the mechanism of action of different antibiotics, and puromycin is a very important antibiotic if you are appearing for GATE papers. Whether it is biotechnology or whether it is life sciences, it is important that you know the mechanism of action of puromycin and other antibiotics. So first of all, it blocks translation by resembling the amino acyl end of the charged tRNA. So option A is correct. However, you should know to which side in the ribosomes it binds and hence blocks translation, and that is A site. So the correct answers are option A and B. All right. Moving on to the next question. factor H, a complement regulatory protein in plasma, binds C3b, and which of the options are correct? So first of all, factor H basically suppresses inflammation, that is mainly common by elevated levels of C3a and C3b. So first of all, it competes with factor B to displace BB from convertase. So if you look at the alternate pathway of complement system, the C3 convertase is C3b-BB. So basically, it competes with this factor B and displaces BB, and it also uses factor I as a cofactor. So basically, it's a factor I mediated C3b cleavage, and hence it decreases the activity of this C3 convertase. Basically, it decreases and hence suppresses inflammation, or even at very worst cases like anaphylaxis or something like that. So factor H is thus a regulatory complement protein in plasma that competes with factor B and also acts as a cofactor for factor I. Okay. Moving on to the next question. In Michaelis-Menten equation, if S is 15 km, that is substrate concentration is 15 km, then the ratio V naught by V max is. So Michaelis-Menten is a very important equation in any biology or biotechnology or life science competitive exams. So the equation, Michaelis-Menten equation is V naught equal to V max into S divided by Km plus S. So V naught by V max equal to S by Km plus S. So that is going to be, S is 15 km, so you just have to substitute these values, Km plus 15 km. So that is going to be, if you cancel all K, this will be 15 by 16, and the answer comes to 0.93, I'll write it here, 0.938. So they have asked to round it off to 3 decimal places. I will safely write it as 0.938. So if you know this equation, this is such an easy question to solve. Moving on to the next question. 5250 base pair long plasmid with 10 negative supercoils would have a linking number of what, considering 10.5 base pairs per turn for the bDNA. So you should know the formula that is linking, that is connecting linking number, twist and width. So L equal to T plus W. So basically we need to find this L. So T is equal to, which is twist, equal to the total number of base pairs, which is 5250, divided by number of base pairs per turn, that is 10.5. So when you calculate this, it comes to 500. And width mainly refers to the number of supercoils, and here it clearly says negative supercoils, so it's going to be minus 10. So now L is equal to 500, which is T, minus 10, which is 490. So if you know this formula, this question is also going to be very simple to solve. And moving on to the last question. The spectrum of a protein obtained using electrospray ionization mass spectrometry, which is ESIMS, is shown below. Two peaks, one at m by z equal to 2960.6 and the other at m by z equal to 3552.5 are marked. The mass of the protein associated with the m by z equal to 2960.6 peak is Watt Dalton, and you need to round off the answer to 2 decimal places. I believe that you all have a fair idea on how ESIMS works and how the spectrum is usually represented. So here clearly in the spectrum, they have given the m by z value for the tallest peak, which is 2960.6, as well as the m by z value of the neighboring peak. So let me start solving this question. Let me take x to be equal to the larger m by z value, which is 3552.5. If I'm not wrong, yes, 3552.5, and y equal to the smaller m by z value, which is 2960.6, right? Yes. So if you have to write the equation for x in terms of m and z, that is mass and charge, since it is the higher m by z value, we write x as m plus z divided by z. Similarly, since y is the smaller value and is the neighboring peak to the highest peak, we write y as m plus z plus 1 divided by z plus 1. I'm not going too much into the detail behind the theory of this, but if you read about mass spectrometer, then these are pretty simple to understand. For the purpose of this video, I'm just sticking on to solving this sum. So when you rearrange these two equations, you get z equal to y minus 1 divided by x minus y. So if you feed in these values, you will have 2960.6 minus 1 divided by 3552.5 minus 2960.6. So if you solve this, you will get the answer as 2959.6 divided by 591.9. So when you do the division, you get the answer to be 5. Alright, so now you need to find the mass of the protein associated with the m by z peak, which is 2960.6. So here we have clearly given y to be equal to 2960.6. So y is equal to 2960.6, which is equal to m plus z plus 1 divided by z plus 1. So if you actually arrange this equation, you will get m equal to y into z plus 1 minus z minus 1, right? And then if you feed in these values, that is 2960.6 into 5 plus 1, which is 6, minus 5 minus 1, you will get the final answer to be roughly around 17763 minus 6. I'm actually avoiding the decimals here just to make it simple, and the final answer here now comes to 17757. Okay, so the ultimate answer is, ultimate answer for the mass of the protein is 17757 Dalton, and that clearly falls in the range here. Yes, hope the question is simple and clear to you. So with this paper, with my experience, I would say that the GATE 2024 Biochemistry XL paper was pretty standard, and I would request all the viewers to level up their preparation in order to ace the upcoming GATE exams as well as any other biology competitive exams. So hope you benefited through this video. Thank you and have a nice day.